Okay, now in this section, I'm going to go into great depth about our old friend Brettanomyces, which is a yeast. Some consider it a spoilage yeast, some consider it the heart of the great Bordeaux, and it's probably both. So I studied Brettanomyces for my master's thesis at Davis, and so I've got quite a bit of information, hopefully some of it fresh and new. I want to start by talking about variability in Brettanomyces. So first, let's look at the high variability in morphology, and you'll see right away what I'm talking about. In these photographs, you can see that sometimes it looks like a mycelium, and other times just like a regular budding cell. And it has this goofy pointed end over here that's called apiculate. And if you just look throughout these, you can see how you just never know what you're going to get. This is light microscopy, and in particular, a mark of Brettanomyces is sort of a rowboat shape that we see. Here's a good example right here. And what's going on, here's a scanning electron microscope blow up. And what's going on is that unlike Saccharomyces, these yeasts only bud to make a daughter cell on the ends of the cell. And so you can see how, depending on the angle you're looking at, you'll either have a sort of a flattened end here, or you'll end up with a pointy end, or sometimes you get pointy ends on both. But this is what's really going on, is you have a row of buds in either case. This would be pointy and this would be flat like a rowboat. Extremely variable in its morphology, that is to say what the cells look like under a microscope. It's also extremely variable in vigor. There's weak strains and strong strains and everything in between. And it varies much more than any other species. And the species here is Brettanomyces lambicus. And it's just crazy how much it varies. Strains can be anywhere between 19 and 21 chromosomes. What organism does that? And then the aromatic expression, partly because of the difference in strains, is quite variable also. All right, now let's start off by talking about Brettanomyces versus Decora. This is a very strange division that exists in mycology. We're talking about the kingdom of the fungi, and Brettanomyces and Decora are classified in completely different families. And the only difference is that Decora produces sexual spores and Brettanomyces doesn't. My personal belief is that these are identical, and it's only the most vigorous strains that have the energy, if you like, to be able to reproduce through sexual spores. Otherwise, I think they're the same thing, and I think it's ridiculous that they have different names. So there's a big debate, mostly between professional mycologists like Dr. Herman Pfaff and Dr. Marty Miller, that I served under in their lab when I did this work, versus practical wine microbiologists like Lisa Vanderwater about what we should be calling it. Lisa always wanted to call it Decora, no matter what it is, because of the possibility that the spores are smaller than the cells, and therefore we have to use a tighter sterile filter. But a mycologist will always go with Brettanomyces until you have proven that you have sexual spores. So Robert E. Logan and I did our theses together, and so what Robert and I were able to photograph were ascospores. They look like helmets. And so there's one right there. And you'll notice that right next to it is a cell that's smaller than the ascospore. And so because of the variability in morphology, this argument about always calling it Decora really doesn't hold water. Here's another example. Here's a whole lot of ascospores. There's a good one right there. There are several others once you know how to look for them. This has got 2, and this has got 3. And if you get them lying just right, you'll get this kind of helmet shape. But here again, we have cells that are just as small as the ascospores, and so the whole thing about filtration is ridiculous. But at the same time, I think the mycologists are kind of out to lunch to insist that these are not the same species, Brett and Decora. They obviously are in all the physiology and morphology, the substrates they can metabolize and so forth. And so I don't really care what you call it. Now, here's a study on the left here that was done by the Australian Wine Research Association, and they were able to find, just in Australia, these 6 different strains, and they're kind of showing where you find them in the wine growing regions. Western Australia, South Australia, Tasmania, the Barossa and the Murray River and the Hunter Valley and so forth. All these different regions have different strains, or sometimes a mix. Like here in the Yarra, there's 2 or 3 different strains. Then similarly, here's some work done in Europe where we have another 6 strains that they found in wine and beer and even tequila. All of this is what's going on throughout Europe. And in particular, you'll note that the Bordeaux strains are almost completely different than the Burgundy strains. This is because Bordeaux and Burgundy don't have a bulk wine business between them because it's illegal and they're different varieties. So you're going to see these differences in expression throughout the wine growing areas of the world. It's really a box of chocolates. You never really know what you're going to get. Now, the aromas and flavors, the most celebrated is 4-ethylphenol, and that is a semi-unique marker. If you have an elevated amount of 4-ethylphenol, then that probably indicates Brettanomyces. But that doesn't mean that that's the aroma. It does have an aroma. It's kind of like an old Band-Aid box or shoe polish. It's really not a defect, and it doesn't smell anything like horse sweat. A little bit more like that is 4-ethyl guaiacol, which has a smoky meat, salami kind of smell to it, so it's a little bit more animalia. These 2 compounds are interesting because they synergize, and as I'll show you later, some red varieties have the precursor ferulic acid for 4-ethyl guaiacol. So even though it's present in smaller quantities, usually it's 4-to-1 in favor of 4-ethylphenol, or maybe 6-to-1. Varies with the variety. Cabernet Franc tends to be high in ethyl guaiacol, this smoky meat character. But the 2 synergize, and so the more you have of one, the more you can smell the other. It's an interesting phenomenon. 4-ethylphenol and 4-ethyl guaiacol are only produced in red wine because the precursor for ethylphenol is fumaric acid, and for 4-ethyl guaiacol it's ferulic acid, and we don't have those in white wine. So white wines with Brett don't smell the same way as red wines do. But there are a lot of other compounds that are, I think, more important in terms of Brett as a defective aroma, and probably the most important is isovaleric acid, which smells like vomit. Then adding to the mix, we have, as I told you in the previous session, we've got volatile acidity. It doesn't produce a whole lot, but it does produce some, and that tends to color the aroma. And then you have, now these are not aromatic compounds. They cause what's called mousiness. That's in the finish of the wine, and you really only taste it after about 30 seconds after you swallow the wine. Brett causes mousiness in wine. We can also get mousiness from these tetrahydropyridines from lactobacillus in juice. And so it's very common. Wines made in the Central Valley, when the truck has to park in the hot sun for 24 hours while it's in line with 100 other trucks at some big winery, then you'll end up getting quite a bit of mousiness out of those, but it only occurs when there's a lot of sugar present. These aromas are also found, or things like them, are formed by Pediococcus bacteria. So Brett and Pediococcus are often confused, and they often occur together. And the classic for Pediococcus is sweat socks, like a gym locker. So here's what I was talking about is these are the precursors, which are then taken through a couple of enzyme steps here that are shown. And what you end up with is 4-ethylphenol and 4-ethylguaiacol and this other thing called 4-ethylcatechol. Only happens in red wine. Now, in my thesis, I explored how clever this organism is. It has a bunch of strategies for causing secondary infection on the backs of other organisms, starting with Saccharomyces cerevisiae primary fermentation. And it's very well equipped to come on after that and then to hide out. So to begin with, it has a very slow growth habit, even under ideal conditions, which are aerobic situation with a nice nutrient-rich agar plate at 30 degrees C, which is its optimum grow point. It really likes heat. It still takes 8 days to get a colony, whereas Saccharomyces, you'll see it the next day. It's very temperature intolerant. It goes into hibernation below 59 degrees, and so you can have it in a cold cellar, but it won't actually show up until the wine is warm. And often a cold cellar, then you bottle without sterile filtration, you'll end up with a Brettanomyces bloom in the bottle. It's very common. It's so good at sequestering itself, and so it'll dig into wood. Then when you go in to steam sterilize or ozonate the barrel, you'll kill off all the cells on the surface, but that layer of dead cells actually protects the organisms behind them in a crack or crevice, for example, between staves. There are some essential nutrients that it needs, in particular biotin and folic acid, that it doesn't know how to make, and they're essential. So it won't grow on its own until some of those micronutrients are present after having bled out from dead Saccharomyces cells, for example, as they die and then lyse themselves and release nutrients into the finished wine. So that's a way that it can keep itself from competing and just show up later on after the wine has been made and other organisms' activity has died down. At that time, it has an incredibly wide ability to use different carbon and nitrogen substrates that other organisms, particularly Saccharomyces, cannot use. I'll go through some of those. Also has peculiar resistance to SO2. I'll talk a little bit about that. A part of my thesis was to isolate 58 strains of Brett, and then I looked at how much SO2 it takes to kill it. And what I used as a definition of kill is I would plate the organisms to get a cell count, how many CFUs, colony-forming units, we found. So like with Saccharomyces, in an active fermentation, you'll have about 10 to the 8th cells per mil. So I grew up all these Brett cultures, and then I added SO2 to see how much it would take to drop the CFU by 6 orders of magnitude. So if I had 10 to the 8th, then if I only found 10 to the 2, I'd call that a kill sensitivity. However, we have since learned with, everybody knows what PCR is now, forensic DNA testing, and we have learned that SO2 does not kill Brettanomyces necessarily, but what it does do is it renders it unplateable, and so we call this viable non-culturable. The joke's on me because it means all that work I did for years was really a complete waste of time because I wasn't really killing the cells. I was just making them invisible. Okay, so let's look at some of these unusual substrates that Brett can utilize, and you'll see how clever this organism is. So for starters, it can actually use ethanol. As long as there's oxygen around, it can utilize ethanol as a sole carbon source for growth. Doesn't need anything else. And it also can utilize proline. Now, proline is what's called a secondary amino acid. It's a ring structure, and it's the most prevalent amino acid in both wine and beer. There's a lot. There's like 3 grams per liter of proline, but Saccharomyces can't utilize it. But lo and behold, Brettanomyces can, as long as there's some oxygen. And then for fermentative carbon sources, of course, you can use glucose and all of the expected sugars, galactose and so forth. But what's kind of weird is that it can use cellobiose. That's a sugar that doesn't exist as a metabolic product in nature, but it's caused by toasting wood. And Brett can use it. And that's the reason that you get more Brett in a newly toasted barrel than you will in a used barrel. So a lot of winemakers like to put their wine in brand new barrels to avoid Brett, but in fact they're encouraging it. So the joke's on us yet again. And then as a sole nitrogen source, proline. It can use all the amino acids, but the fact that it can break into the ring of proline and use it both as a sole carbon source and as a sole nitrogen source is kind of mind-blowing. Okay, so here's what I was talking about. These are the phases that we like to discuss. I call them 0, 1, 2, and 3, but what's shown here, they're calling them A, B, C, and D. So the first one is what you get prior to the onset of alcoholic fermentation by Saccharomyces. So that's a period where we might call it cold soak before the Saccharomyces had a chance to take over. Then here's the Saccharomyces doing its thing and then dying off. And after that, we get the malolactic starting. And then after that, we get the post-secondary fermentation of organisms like Brett and Acetobacter and Pediococcus, and also a lot of beneficial organisms. I'm going to talk about those in a minute. Most of what we get in phase D is indigenous microbiome from the cellar, and much of it is not only not spoilage, but it's beneficial in outcompeting particularly Brettanomyces and Pediococcus, that if we sterilize the wine and take away the beneficials, then we will get a very strong Brett infection, much stronger than we would get if we hadn't messed with Mother Nature. So, for example, in sulfite-free wines, we really don't have problems with the Brettanomyces because everything is growing like crazy in those wines and protecting the wine. So let me tell you a story of when I was working with Enadev, and we were first in the 90s experimenting with microoxygenation. People were concerned that minute amounts of oxygen would allow the Brett to metabolize, for example, ethanol, and that we would encourage Brett growth. And so we wanted to do an experiment to see whether that was true. And what we did is we took some 12-inch stainless steel pipe, and we made these little tanks that were 8 feet tall, had 12 of them, and we filled them all with Merlot. This is in France. And we inoculated 6 of them with Brettanomyces, and the other 6 we didn't inoculate. Now, to have it be a rigorous scientific experiment, we flash pasteurized the wine, high temperature, short time, as we put the wine into these little tanks. And so then we had 6 different regimens of microox with and without this inoculum of Brett that we had introduced. Well, we had to abandon the experiment. Now, people start to freak out when there's more than 500 cells per mil of Brett. We got 10 to the 7th in all 6 inoculations, and that's when I realized that if you kill off everything in the wine, the Brett goes insane. So it's really like a hospital disease, an opportunistic pathogen, if you like. And draconian cellar procedures like sterile filtering or Velcorin, unless you're going directly into the bottle. If you use sterile filter wine and put it back in a barrel, then you're really asking for trouble. Now, this slide was provided by my friends over at Inardis, and they have a material, as do some of the other guys, but I'm going to use their slides here, and it's made out of chitosan, which is what the shells of crustaceans are made out of, and they have a material that they make from aspergillus spores, and it's very effective in suppressing the growth of Brettanomyces and other bad actors. This is what the aspergillus mold, that is that black stuff that you've seen many times. That's the source of this stuff. That's what it looks like under a scanning electron microscope. Their process kind of roughens the edge of it, and they think that makes it more effective because what it does is it physically goes in and incorporates into the Brett cell. Stab micro M is for musts, so that's fine if you want to use it in a must, but they have almost the same name for the stuff you do use in wine, which is just stab micro. And why on earth they give these two practically the same name is beyond me, but watch yourself. It's also effective against Pediococcus. You can see here that 10 grams per liter is knocking the Pediococcus way down. It's effective against Acetobacter. Here's your treated wine on malic acid. So it's good at preventing malolactic. On the other hand, if you want to have a malolactic, you better not put it in the barrel until after you get your completed ML. But for whites, where you're trying to suppress ML, it's really great. Here's another experiment where we have Acetobacter. In the control wine, you get an elevated level of acetic, whereas the treated wine keeps it down. So it's pretty cool stuff as generally suppressing microbiology and the three bad actors of Brettanomyces, Pediococcus, and Acetobacter. I want to mention that there are other chitosans out there. This was the first one introduced to the market. It's called No Brett Inside, and you get it from Scott Labs. And then this is Kill Brett. That's from La Motte Abiette, which is the Bucher Vaseline materials side. This is where Eglantine Chauffeur hangs out. And I haven't played around with these three products long enough to really be able to tell you which is best. You're going to have to figure it out for yourself. But they all seem to work. They've been a real lifesaver for me, particularly when I have varieties that are prone to either Brett or Acetobacter, like the highly colored reds that I was telling you about before. So this is what I use as an alternative to SO2 to control Acetobacter in high pH, highly colored reds. So then we can also adapt the reverse osmosis filtration to remove Brett character, only it's not going to work quite the way it does for acetic acid because these compounds are a little bit bigger, and so we're going to have to use a looser reverse osmosis filter, and then that permeate, instead of going through an anion resin, we'll go through a broad spectrum adsorptive carbon here and then be returned to the wine. And unfortunately, your wine is going to take a hit. You're going to have some flavor stripping with this looser RO membrane, and you may have to do some creative blending after the process, particularly in a light wine like a Pinot Noir. It doesn't have the tannic structure to hold up, but many people have had good success with this methodology. I will say that I don't think it's the composition of Brett that causes wines to be Bretty. When we talk about good Brett and bad Brett, I think it's got all to do with the macromolecular structure, the colloidal structure in the wine. And I like to use microoxygenation to create a refined colloidal structure that incorporates Brett. The combination of that and trying to nurture a healthy, beneficial microbiome is the strategy I use, and I end up with little tiny amounts of Brett that, to me, very much enhance the quality of the wines I've got. And I learned something recently where I was tasting with a group including Rob Davis from Jordan, and we were tasting the 76 cab. It's the first cab that he made under the tutelage of Andrei Telachev. And he pulled out a 750, and Tom Jordan had always insisted that they sterile filter the wines. The wine was in very good shape for being so old, but it hadn't really developed very much. And then he pulled out a Magnum and he said, well, Andre and I decided we wanted to do some Magnums and we couldn't fit the bottles onto the bottling line. So we weren't able to sterile filter the wine. But Andre said, oh, what the heck, let's just take a siphon hose and we'll bottle right out of the barrel. And that's what they did. And this wine, to me, was vastly superior. There was nothing wrong with the Jordan. It wasn't oxidized or anything, the regular 750, but these were really like a great Bordeaux. And that's when I learned that a lot of the development that we expect in a bottle is really microbiology. That was a real eye-opener to me. Okay, now I'm just reminding you, you've seen this chart before, but just remember that the tight RO, that's going to be about 80 Daltons or so. That's what you use if you want to remove acetic acid or ethanol. But if you want to get at these volatile phenols down here, then you have to use a loose RO in the range of 120 to 150 Daltons. And you are going to take a flavor hit there. And then what we've learned recently is that we'll be able to remove smoke taint. When we get to very loose RO, we might be as much as 500 Daltons if we want to take out smoke. And then the wine, of course, really does take a hit, and we need to go find some Petit Verdot from someplace or something to give it more body. And also perhaps some aromatic woods like cherry and acacia to supplement the fruit in the nose of those wines post-treatment. And coming up next, sulfite-free winemaking. We now have all of the elements under our belt to be able to talk about making wine without sulfites. I do quite a bit of this, and it works better than you might think.